The human gut microbiome protects the body against pathogenic invaders, supports the immune system, and assists in regulating metabolism. Other benefits include enhanced cognition and mood, increased nutrient absorption, reduced hypertension, and alleviated arthritis. The gut microbiome is an evolving environment, and the composition of bacteria can be affected by diet, disease, and environmental stressors. Probiotics are live bacteria cultures naturally produced in fermented food products. Probiotics introduce beneficial bacteria into the GI tract. Prebiotics, in contrast, are non-digestible food ingredients that stimulate the growth and activity of beneficial bacteria in the gut. Prebiotic ingredients are not hydrolyzed in the upper GI tract, so they reach the colon and provide nutrients to the gut bacteria. Onions, chicory root, garlic, and apples are all considered high prebiotic capacity foods. Onions, chicory root, and garlic contain dietary fibers, specifically inulin and fructooligosaccharides, that provide necessary nutrition to intestinal flora. The objectives of this experiment include to determine the effectiveness of various prebiotics on the growth of beneficial bacteria in the human intestinal microbiome, and to evaluate which natural products can be incorporated into our everyday diet to promote health and well-being.
This experiment will examine the symbiotic effects of lactobacillus, a common probiotic supplement, when exposed to 4 prebiotic ingredients, which are apple, onion, garlic, and chicory root. First, we plate the prebiotic capsule and grow the culture, proceed with dilutions until a suitable optical density reading is obtained, which ensures a countable range of colonies. Then we mix 1/10 of a milliliter of the bacterial culture with 100 milliliters of the 4 prebiotic solutions, plate on MRS agar plates, cover and incubate at 37 degrees Celsius for 48 hours, proceed with enumeration counts and colony health observations, and finally repeat twice and average data from all 3 trials.
The first study that we found that tests the effectiveness of prebiotics on bacterial growth is shown in Figure 1. In this study, the 2 main prebiotic compounds found in onion, garlic, and chicory root were added to a stimulated human gut. The growth of several gut bacterium was recorded before, during, and after the inulin and FOS were added to the simulator. The next study that supported our experiment was a double-blind experiment done on 30 obese women who either received a prebiotic pill containing a 50-50 mix of inulin and FOS or a placebo. Figure 2 shows the bacteria count in the women's fecal samples. In addition, the overall health of the women who took the prebiotic versus the placebo was compared.
The second study that we found that tests the impacts of probiotics on bacterial growth can be seen in Figure 3. The main prebiotic component found in apples is pectin, a complex plant heteropolysaccharide. Pectin was mixed with a nutritive broth called CAP and was then added to lactobacillus on several growth medias. The cell count of the growth medias was recorded before and after the CAP broth was added. As a result of the CAP broth, an increase in viable cell count and controlled pH levels was recorded.
Figure 1 shows the effect of the prebiotic compound inulin and FOS on gut bacterium. The results of the study show prebiotics aid in the growth of gut bacteria. The growth of bacteria, including lactobacillus, was observed during the administration of the prebiotic and after the prebiotic was no longer being administered, indicating that prebiotic fibers may aid in bacterial growth for prolonged periods of time. Figure 2 shows the growth of bacterium in human subjects after taking prebiotic pills containing FOS and inulin. The women who took the prebiotic saw significant growth in the bacteria in their fecal samples compared to the women who received the placebo. In addition, the women who took the prebiotic saw a slight decrease in BMI and overall metabolic activity increased slightly.
As can be seen in Study 3, the cultivation of lactobacillus in different types of media containing apple peels as the sole carbon source resulted in intense bacterial metabolic activities, indicated by high viable cell count. Apple peels also presented positive prebiotic scores towards all the tested lactobacilli strains, including a fermentation activity. Clear increase in the cell count when CAP broth was added was also observed. This concludes that apples are also an effective prebiotic because they include an active ingredient that fosters viable cell growth.
In conclusion, while all 4 prebiotic foods in question promote bacterial growth, evidence indicates that inulin and FOS fibers are the most significant active ingredients in nourishing intestinal flora. Therefore, because chicory root has the most concentrated content of inulin fiber, chicory root is the most effective product in promoting the health of lactobacillus colonies in the gut microbiome.